All right, so we are going to talk about mammalian or animal reproduction, and specifically, we're going to be talking about mammal reproduction, not all animals. These are animals that sexually reproduce and not the animals that lay eggs, is what we're going to be talking about here. So, all mammals have sexual reproduction. They have haploid gametes by meiosis, and the control of the gamete production is dependent upon hormones. Mammals are viparious or viviparous except for monotremase. These are talking about placental animals that retain their young in utero, not the ones that lay the eggs outside of their body. Most mammalian offspring require large amounts of prenatal care. Males play a very small role in parental care in most cases. Why is it not going on? What did we do here? Oh, that's... I don't... hang on. Slow down. I don't know why it's not working appropriately. There. All right, so what we're going to talk about is the human reproduction system. So we'll talk about the female reproductive cycle, which involves periodic releases of a mature ovum. This is called ovulation. Most female mammals have an estrous cycle, and females are sexually receptive to males only at the time of ovulation or in estrus. Primates, including humans, all have menstrual cycles. Sexual differentiation is determined genetically by the presence of a Y chromosome at the 23rd pair. A Y chromosome indicates that you have a male. 2 X's indicate you have a female species. A developing embryo has Wolffian ducts and Mullerian ducts. Wolffian ducts develop into the male epididymis, vas deferens, and accessory organs. The Mullerian ducts will develop into the vagina, uterus, cervix, and oviducts. In males, genes located in the Y chromosome initiate differences of epithelial cells into the Sertoli cells. Sertoli cells produce anti-Mullerian hormones. This causes the deterioration of the Mullerian duct. This is a defeminization process. Mesenchyme cells differentiate into Leydig cells. Leydig cells produce the androgens, or the male sex steroids, at this point. The androgens, which are testosterone and 5-dihydrotestosterone. Testosterone is responsible for the development and the differentiation of the Wolffian duct, and dihydrotestosterone is responsible for the masculine development of external genitalia. The human reproductive system. Females have the 2 X chromosomes. They have an absence of testosterone, which leads to the feminization or default condition. All mammals, regardless of sex, begin life with primordial undifferentiated gonads. If there are no androgens present for the Wolffian ducts to develop, they will degenerate. The Mullerian ducts would then develop due to the secretion of estradiol, which is a different hormone. Gonads would then become the ovaries, vagina, uterus, and cervix. So basically what it comes down to is if this testosterone is present or not present. If the androgen or the testosterone is not present, the Wolffian ducts will not develop. Therefore, the Mullerian ducts would develop. There are developmental disorders. Some people end up with a XO syndrome. They have 1 X chromosome and 1 O, or non-chromosome, essentially. This is an infertile female. We have people that have 2 X's and a Y chromosome. This is referred to as Klinefelter's syndrome. They have testes development. They are considered male, but they are sterile. We also have congenital and adrenal hyperplasia. This is where you have 2 X embryos. I don't know what happened there. In the ovarian development, and it overproduces androgens and the mascularization of the genitalia. They develop Wolffian ducts and Mullerian ducts, but they have no Sertoli cells. At puberty, they end up getting secondary characteristics. So this issue has both male and female characteristics. This is just looking at what happens here with the germ cell going through meiosis 1, meiosis 2, and getting the spermatozoa, and this all takes place in the testes. The male reproductive organs are the epididymis, which is for sperm storage. The vas deferens conducts the sperm to the prostate. The seminal vesicle, the prostate, the bulbourethral gland, and the urethra and the penis. You will need to know all of these reproductive organs for the exam. When we look at the human male reproductive system, this is where everything is located. You will not be asked to identify them, but you will be asked to know their role. So this is the sperm. I think on your study guide it asks you to identify the parts of the sperm as you look at it. It does come with its own mitochondrion and the centriole in here. The human female reproductive system, the ovaries develop more slowly than the testes because they develop after the presence of the androgen is not there. Ovaries contain microscopic ovarian follicles. Each follicle contains a potential egg cell called a primary oocyte and smaller granulosa cells. The granulosa cells secrete estrogen. Primary oocytes begin meiosis but are arrested or stopped in prophase 1 before birth. There are a finite number of oocytes. Therefore, there are a finite number of potential eggs that a female is born with. At puberty, granulosa cells proliferate and secrete estrogen. This triggers the first menstrual cycle, and it stimulates secondary female characteristics. After puberty, the oocyte completes the first meiotic division. However, the first meiotic division is very uneven. It produces a large secondary oocyte and small polar bodies. If you remember, we talked about this during sexual reproduction. The secondary oocyte begins meiosis 2 and is arrested in metaphase 2. Fertilization stimulates the continuation, but only fertilization takes place in the large secondary oocyte, not the small polar bodies. With the menstrual cycle, we have the fetal development here. This is at the very beginning. Then those oocytes are arrested right here in prophase. Then once they reach puberty, the ovulation occurs. You get the secondary oocyte, which is the large one that goes through meiosis 2, which is the egg, and you get the polar body that gets developed here. As it goes through, then it stops and arrests again at metaphase 2 until fertilization takes place. When fertilization takes place, it's going to take place over here in the ovum, and the secondary polar bodies will not be fertilized. Development of mature graphene follicle occurs approximately every 28 days during the menstrual cycle. Follicles remain in the ovary after ovulation and develop in the corpus luteum. The corpus luteum secretes progesterone, again, which is a hormone which stimulates the proliferation or the production of endometrium. And the endometrium is the lining of the cervix. Usually this is a short-lived cycle unless the oocyte, the mature oocyte, gets fertilized. When the corpus luteum dies, there's a loss of progesterone, and this results in the shedding of the endometrium, which causes that cycle. So this is just a chart showing what happens with this, and this is all regulated in the hypothalamus gland within the brain. So you can see the menstruation cycle, the proliferation, ovulation, and moving through every 28 days that this happens. Now what we have to look at are the different phases here. We have the primary follicle that develops into the follicle. We have a mature follicle and the secondary oocyte. Then if fertilization takes place, it's going to take place over here, but a follicle gets released into the fallopian tube each time. But fertilization occurs, then it continues. If it doesn't occur If fertilization doesn't occur, it just gets reabsorbed back into the fallopian tubes. If it does occur, fertilization occurs, it continues along and gets fertilized, and it gets the cleavage furrow and continues fertilization. After fertilization, the developing embryo will produce a human chorionic gonadotrophin, which is again another hormone. It maintains the corpus luteum and prevents menstruation. This keeps levels of estrogen and progesterone high. So this is a hormone that is tested for in pregnancy tests. So this is why whenever female mammals do not get their next menstrual cycle, it is often an indication that they have a fertilized embryo. So when we look at the locations here, this is the ovary where it begins in the fallopian tube that we just looked at. And this then goes into the uterus and the uterine lining and the cervix, which is in here. So you can see everything that's associated with the fertilization process. Now we're getting into what happens after this fertilization. This is developmental biology. So this is kind of the second half. After fertilization, which is the union of the male and female gametes, it consists of 3 events. The sperm penetrates the membrane fusion, the egg is activated, and then the nuclei of both the sperm and the egg fuse. Then development involves growth of that nuclei and those cells, growth in numbers of cells, differentiation between what the cells are made for, and then the specialization of those cells to perform specific functions. So fertilization, a sperm must penetrate into the plasma membrane of the egg for the membrane fusion to occur. The egg is then enveloped by one or more protective coatings. This is what's called the zona pellucida in mammalian eggs. Then we have, once the membrane fusion activates the egg, the sperm makes contact with the egg's plasma membrane. Excuse me, sorry. So, changes in the membrane of the potential egg prevents other sperm from fusing with the eggs. Enzymes from the cortical granules remove sperm receptors. This prevents more than one sperm from fertilizing the egg, essentially. Sperm triggers the egg to complete meiosis. Remember that the oocyte has been arrested in metaphase 2, so this will restore the diploid state from a haploid sperm and a haploid egg, and we continue. There are proteins on the surface of the acrosome that bind the receptors of the membrane. This is a very specific match that secures species specificity. Meiosis is where we have the formation of the male and female gametes. Fertilization then happens. Then we get the cleavage, which is the rapid division of the cells. Gastrulation, which is where the cell division slows, but the cells go through extensive rearrangement. The ectoderm, the mesoderm, and the endoderm. Then we go through organogenesis, which is the development of specific organs. The cleavage, which is the rapid division of the zygote into larger number of smaller and smaller cells called blastomeres. There is no overall increase in the size of the embryo. We're increasing numbers of cells only. The animal pole and the vegetal pole. The blastomeres of the animal pole form external tissues of the body. The blastomeres of the vegetal pole form the internal tissues. So this is what we look like, and you will need to be able to label these parts on the exam. A fertilized egg, first cell stage, second, fourth, then the 32, 64, but getting into the blastula and the gastrula stages will be important. You will have to label these on the exam. So a hollow ball of cells, or the blastula, contains a fluid-filled cavity called the blastocele, and that is right here. Okay, you can see the animal pole, the vegetal pole. Remember what the animal pole does and what the vegetal pole develops into. External, internal. Don't forget that. In bilateral phyla, all organisms have developmental stage involving three tissue layers: the ectoderm, which develops as the outside, the mesoderm stays in the middle, and the endoderm, which is the innermost layer. In protostomes, the mouth develops first. These are worms, arthropods, and mollusks. The column is formed by hollowing out a solid mass of cells that makes up the mesoderm. In the middle, this is the body cavity essentially. It undergoes a spiral cleavage, and this determinate cleavage is the fate of cells is determined early. In deuterostomes, the mouth forms second. These are chordates and echinoderms. The mouth forms from the opening on opposite sides of the blastopore. The blastopore is an opening to the outside, first opening in development. The radical cleavage, or the rational cleavage. Then the interdemental development, as the fate of cells is determined by the interactions between other cells. Deuterostomes evolved from protozoans approximately more than 500 million years ago. So here's a chart of kind of what happens. We get, these are Sporullian protozomes and these are deuterosomes. So you can see how they start off virtually the same, but during the embryonic cells, things change. And then the blastopore becomes the mouth, and here the blastopore becomes the anus. Then we have the mesoderm levels that get developed in here, and after that they become the same. Okay? So the pattern of development is dependent upon the amount of yolk when we're talking about the blastula of mammals and birds. So we're talking about egg cells, and these inner masses of egg cells, or we consider it the yolk, if you will, when you think of mammalian cells, but we think of eggs, the yolk inside. That mass of cells determines the pattern of development. Egg yolk contains proteins or huge stores of messenger and transfer RNA and ribosomes. This powers the egg during the cleavage process, that rapid division, and morphogenic determinants decide what the cell becomes. In eggs containing moderate or little yolk, the cleavage occurs throughout the egg, and you have a holoblastic cleavage. Eggs containing large amounts of yolk have a cleavage or incomplete meroblastic cleavage will occur. Holoblastic cleavage gives eggs without very much yolk. It results in a more evenly sized cell. The cleavage furrow extends throughout. Meroblastic cleavage furrows cannot make it through the yolk, and you have incomplete. In birds and reptiles, the cleavage is restricted to the region of the concentrated cytoplasm. So meroblastic cleavage we have here. We have the yolk, and we get that meroblastic, remember, we're going to get that uneven cleavage furrow. Compaction at the 8-cell stage. These are where we have the outer cells which develop into the trophoblasts, which becomes part of the placenta. The inner cell develops into the embryo, and random is no one cell is destined to be an inner or outer cell. It is very random. Blasticle forms by the secretion of the fluid by the trophoblast cells. Now we get into gastrulation. This is the migration and rearrangement of cells of the blastula. This results in the formation of the endoderm, mesoderm, and ectoderm. The endoderm, and please know this information for the exam also, the endoderm becomes the internal organs. The mesoderm becomes the bone, heart, blood vessels, muscles, connective tissue, and gonads. The ectoderm becomes the skin and the nervous system. You will be asked to remember that. So during gastrulation, the cells move along the gastrulation, undergoing a variety of cell shape changes. Cells are attached tightly to the junctions of the cells, will move then as sheets. You can see how the cells are moving as sheets. Here are these migrating cells. Okay, the invagination means the sheet dents inward. So you can see the sheet denting inward right here and folding over upon itself. In mammals, embryo develops from the inner cell mass. A thickening of the cells forms a primitive streak. This primitive streak is what's caused right here. That is where that fold is going to occur. That's the cleavage, essentially, that we're going to get there. In the middle of the primitive streak, the cells will sink inwards, forming a primitive groove. Cells entering the sides of the primitive groove become the mesoderm and the endoderm. Cells that stay outside become the ectoderm. So gastrulation, this is what happens. This is in pictures. The inner cell mast and the trophoblast. We have the formation of the yolk sac, those proteins that are high in those RNA molecules. We then get the ectoderm and the endoderm separated. And then what happens? The primitive streak starts to form, and some of those come out and start to form the mesoderm in the middle. Okay? So, extraembryonic membranes. Okay? As an adaptation to terrestrial life, the embryos of reptiles, birds, and mammals develop within a fluid-filled amniotic membrane, or an amnion. The chorion is the fetal contribution to the placenta. The yolk sac plays a critical role in the nutrition of birds, reptiles. It is present in mammals, but not nutritive. Allantois in birds fuses with the chorion and facilitates the gas exchange. In mammals, it contributes blood vessels and the developing umbilical cord. So we can see this is a chick embryo. What you will notice is at this stage of embryonic development, all mammals, all animals look alike. So we have the chorion, the amnion, the yolk sac, and the allotonus down here. Now the allotonus will become something different, but this is a chick embryo. We're going to look at a mammal embryo. We have the chorion, the amnion, the yolk sac, the umbilical vessels here that have been developed from the allotonus down here. So you can see the yolk sac is smaller because it's non-nutritive down here, because the nutrients are being fed by the mother. In the egg, the yolk has to feed the young chick because they're external. So you have the maternal blood that are feeding the mammalian embryo here. But notice we still have all the same constituents and the same look. Neutralation is the formation of the nervous system. The first organs that develop in the embryo, the folds of ectodermal tissue come together, forming the neural tube. The neural tube has to close off, and the neural tube becomes part of the spinal cord. So the neural tube formation is what forms next, and we get the plates and everything here that form these neural or nerves or neurons. And then what happens is it creates the fold over. We get the neural tube being formed. Cell migration now. Cells will migrate to different parts of the embryo to form distinct tissues. Example here, cells of neural crests form the sense organs, the ears, the eyes, the nose, the mouth. Organogenesis is the formation of the organs in their proper locations. This occurs by the interaction of cells within and between the three germ layers. This follows rapidly on the heels of gastrulation. At some point, every cell's fate becomes fixed. These cells are then determined. This is cell determination. All of the cells in the animal's body, with the exception of a few specialized ones, have now lost their nuclei, and they contain the same complement of genetic information. To a large degree, a cell's location in the developing embryo determines its fate, but it is only true up until a certain stage of development. Ontogeny, or oncogenics here, if we want to think about that, re-encapsulates phylogeny. So in the early 1800s, Van Baer noted that early embryos or members of the phylum and subphyla appear fairly similar. So this is when he started kind of the Linnaean scheme, the class, order, family, genus, species of human development. Developmental patterns of more recently evolved groups have been built on more primitive patterns. So we've had people that have, surmised different things about evolution based on these developmental stages. When you look here, look at a salamander, a fish, and a tortoise, and you look at the embryonic development stages, you can see at the very beginning there's a lot of the same. But we look later and we see lots of developmental differences. Then you look at a chicken and a human, and you can kind of see at the very beginning lots of embryonic similarities, but then we start to move and differences come about. That is all. I will see you guys shortly.